Hello colleagues and students, I'm Jin Yu from the Chinese University of Hong Kong. I would like to share our latest paper published in Gut, Lactococcus lactis, HkyuLL suppresses colorectal tumourigenesis and restores gut microbiota through its generated protein Alpha-mannosidase. The gut microbiota plays a crucial role in colorectal cancer (CRC). In CRC patients, their gut microbiota is featured with significantly decreased abundance of beneficial probiotics. We previously reported several CRC-depleted probiotics, such as Lactobacillus glinarum and Eusebio intestinalis, could prevent colorectal tumourigenesis in mice. Therefore, our findings imply that bacteria depleted in CRC are potential prophylactics of CRC development. Through metagenomic analysis, we identified that Lactococcus lactis is one of the top depleted bacterium in CRC patients from our in-house cohort, as well as in 2 validation cohorts.
Lactococcus lactis is a gram-positive lactic acid-producing bacterium. L. lactis is considered safe for human consumption, and it has been widely used in fermentation of dairy products such as cheese and yogurt. To this end, we investigated the role of L. lactis in CRC. We first isolated L. lactis from healthy human stools. After a series of selective culture, we successfully isolated a new strain of L. lactis termed HkyuLL10. By whole genome sequencing, we confirmed that the genome sequence of HkyuLL10 is different from other L. lactis strains. To test the effect of L. lactis HkyuLL10, we gave this bacteria to APC-mean plus mice, which is a transgenic mice model of CRC. Our results clearly showed that the colon tumor number and tumor size after HkyuLL10 treatment is much smaller than the controls. For validation, we established another CRC mice model by using carcinogen AOM and DSS. Similarly, HkyuLL10 treatment also decreased tumor number and tumor load in a column of AOM DSS treated mice. We also investigated the impact of HkyuLL10 on the gut microbiota by fecal metagenomic sequencing. Microbial communities in HkyuLL10 treated mice were significantly different from the control mice. In particular, probiotics such as Lactobacillus johnsoni and Akkermansia muciniphila were enriched after treatment. These suggested that HkyuLL10 could influence the overall microbiota and increase abundance of gut probiotics. For in vitro experiments, we found that the conditioned medium of HkyuLL10 could reduce the viability of human CRC cells. However, the inhibitory effect of conditioned medium were completely lost after heating. And this suggested that the anti-CRC molecules in HkyuLL10 are proteins. We next separated the conditioned medium into different molecular weights. Notably, only HkyuLL10 conditioned medium with over 100 kilodalton could suppress CRC cell growth. This implies that HkyuLL10 secretes large anti-tumorigenic proteins against CRC.
To identify the functional protein of HkyuLL10, we performed silver staining and mass spectrometry on the conditioned medium with different molecular weights. And we identified that alpha-mannosidase is the functional protein of HkyuLL10. Alpha-mannosidase is an enzyme for gut bacteria to degrade dietary glycans. We found that this enzyme could markedly inhibit the growth of both human CRC cells and tumor organoids derived from human CRC patients. For validation, we generate a mutant E. coli strain that expresses alpha-mannosidase. Compared to its wild-type control, the conditioned medium of this mutant E. coli could decrease the viability and proliferation of human CRC cells. This mutant E. coli also reduced the growth of organoids derived from CRC patients. Finally, we examined the effect of alpha-mannosidase in xenograft mice with 2 different human CRC cell lines. And this enzyme consistently inhibits tumor growth in both CRC xenografts. Altogether, Our in vitro and in vivo results confirm the protective role of alpha-mannosidase against CRC.
In summary, the probiotic Lactococcus lactis is depleted in CRC patients from multiple cohorts. We successfully isolated a novel L-lactis strain, HkyuLL, from healthy human stools. This new strain could inhibit colorectal tumourigenesis and restore gut microbial dysbiosis in CRC mice. HkyuLL exhibits anti-CRC effects by secreting protective protein alpha-mannosidase. Our study therefore elucidates the potential of L-lactis HkyuLL as a prophylactic of CRC, and these findings must facilitate the possibility to use probiotics for CRC prevention. Thank you.